Welcome. Today, we're diving into the fascinating world of emotions and the brain, specifically exploring the concept of basic emotions. For decades, psychologists have debated whether emotions like fear, anger, happiness, sadness, and disgust are fundamental, distinct categories. The central question revolves around whether there are unique neural bases that are both specific and characteristic for each basic emotion. This debate has profound implications for how we understand the brain's organization and the nature of human emotional experience. Throughout this exploration, we'll examine evidence from neuroscience research, including neuroimaging studies, lesion research, and studies of emotional awareness. This journey will help us understand whether the concept of basic emotions holds up under scientific scrutiny and what modern neuroscience tells us about the emotional brain. Basic emotions represent a fundamental concept in psychology and neuroscience. But what exactly are they, and how do they differ from other emotional experiences? Basic emotions are proposed to be innate and universal across all human cultures. They are associated with specific facial expressions that people recognize regardless of their background. These emotions are also linked to distinct physiological responses in our bodies and are believed to be connected to dedicated neural circuits in our brains. Researchers have identified several core basic emotions. This emotion wheel shows 8 commonly recognized basic emotions: anger, anticipation, trust, fear, surprise, sadness, and disgust. But the fundamental question in neuroscience is whether each of these basic emotions has a dedicated and identifiable neural basis in the brain. This leads us to the core question that drives much of emotion research. Does each basic emotion have a dedicated neural basis in the brain? Scientists investigate this question by examining whether specific brain regions are consistently activated for each emotion across different people and situations. For example, researchers have proposed that the amygdala is specifically involved in fear processing, the insula in disgust responses, and the anterior cingulate cortex in sadness. However, as we will explore in the following sections, the evidence is more complex than these simple one-to-one mappings suggest, leading to ongoing debates in the field. Understanding basic emotions and their neural foundations remains one of the most fascinating and challenging areas of neuroscience research. Neuroimaging techniques have revolutionized our understanding of how emotions are processed in the brain. Technologies like functional magnetic resonance imaging, or fMRI, and positron emission tomography, known as PET scans, allow researchers to observe brain activity in real time as people experience different emotions. These neuroimaging studies have identified specific brain regions that consistently activate during particular emotional experiences. The amygdala, a small almond-shaped structure deep in the brain, shows strong activation during fear responses. For disgust, researchers have found consistent activation in the insula and globus pallidus. The insula processes internal bodily sensations, while the globus pallidus is part of the brain's motor control system. Anger activates the lateral orbitofrontal cortex, a region involved in decision-making and impulse control. Meanwhile, the mesial prefrontal cortex appears to be involved in processing all emotions, suggesting it plays a central coordinating role. Now let's examine a detailed brain imaging study that shows the complexity of emotion regulation networks. This diagram illustrates how multiple brain regions work together during emotional processing and regulation. However, neuroimaging research faces significant limitations. Meta-analyses that combine results from multiple studies often yield inconsistent findings. This inconsistency has led some researchers to question whether basic emotions truly have distinct neural signatures. The main problems include variations in experimental design between studies. Different researchers use different stimuli, different tasks, and different analysis methods. When these diverse studies are combined in meta-analyses, the variations can obscure genuine neural patterns. Additionally, emotional experiences are inherently complex and multifaceted. A single emotion like fear can involve multiple brain networks working simultaneously, making it difficult to identify one specific neural signature. This complexity challenges the simple one-to-one mapping between brain regions and emotions that early neuroimaging studies sought to establish. While neuroimaging studies provide valuable insights, they have important limitations. To truly understand how emotions work in the brain, we need evidence that goes beyond correlation to establish causation. Lesion studies offer a powerful alternative approach. These studies examine patients who have suffered specific brain damage, allowing researchers to determine which brain regions are actually necessary for emotional processing. Let's examine a classic example. The amygdala, a small almond-shaped structure deep in the brain, has long been associated with fear processing. But lesion studies provide the crucial evidence that it's actually necessary for fear recognition. When patients suffer damage to the amygdala, they show profound deficits in recognizing fearful facial expressions and responding appropriately to threatening situations. This provides causal evidence that the amygdala is necessary for normal fear processing. Beyond lesion studies, researchers also investigate the relationship between basic emotions and conscious awareness. A fascinating question emerges. Can emotions be processed automatically without conscious awareness? Research reveals that emotions can indeed be processed automatically and unconsciously. For example, fearful faces presented too briefly for conscious recognition can still trigger amygdala activation and physiological fear responses. This suggests that basic emotions have rapid automatic processing pathways. These findings from lesion studies and consciousness research provide crucial evidence that goes beyond simple brain imaging. They demonstrate that specific brain regions are necessary for emotional processing, and that emotions can operate through rapid, automatic pathways that don't require conscious awareness. The traditional view of brain function assumes a simple one-to-one mapping between brain regions and their functions. Each area was thought to have a single, dedicated purpose. This view suggested that the amygdala only processes fear, the visual cortex only handles vision, and so on. Each brain region had its single, specialized job. However, modern neuroscience reveals a more complex picture. The concept of pluripotentiality suggests that a single brain region can serve multiple functions, depending on the neural network it participates in. Let's examine how this works with the amygdala. Rather than just processing fear, the amygdala can participate in different neural networks, each giving it a different functional role. The key insight is that the amygdala's function depends on which other brain regions it's connected to at any given moment. Different network patterns create different functional outcomes. For example, when the amygdala connects with memory regions like the hippocampus, it helps form emotional memories. When it connects with attention networks, it helps direct focus to important stimuli. This network perspective is demonstrated beautifully in fear processing. The amygdala doesn't work alone. It's part of a complex circuit involving visual areas, memory systems, and motor regions. This pluripotential view revolutionizes our understanding of brain function. Instead of asking, "What does this brain region do?" we now ask, "What does this region do in this particular network context?" This network-level perspective moves us beyond simple brain maps to understand the dynamic, context-dependent nature of neural function. It shows us that the brain is far more flexible and adaptive than previously thought. Emotions are incredibly complex phenomena that involve multiple interconnected components working together. Understanding emotions requires examining various types of data and features across different domains of human experience. When researchers study emotions, they examine multiple components simultaneously. Each component provides unique insights into how emotions are generated, experienced, and expressed. This diagram shows how emotional processing flows through different brain regions, from the initial event through the limbic system, cerebral cortex, and brainstem, creating both feelings and thoughts that lead to responses. Researchers collect data across multiple domains: behavioral responses, physiological changes like heart rate and skin conductance, visual cortex activity, eye movement patterns, facial expressions, and brain activity measured through fMRI and EEG. Now let's examine how different emotions can share common features while also having unique characteristics that distinguish them from one another. All emotions share certain common features. They all activate the limbic system, trigger autonomic nervous system responses, involve facial muscle activation, and affect attention and memory processes. However, each emotion also has unique features that distinguish it from others. These include specific brain activation patterns, distinct facial expressions, different physiological signatures and unique behavioral tendencies. Understanding emotions requires examining functional brain connections and how different brain networks communicate during emotional experiences. Modern brain imaging reveals that emotions involve networks of brain regions working together. These networks show dynamic communication patterns that change based on context and require integration of multiple types of data. Future research should focus on integrating multiple types of data simultaneously, conducting network-level analyses of brain function, and mapping features across different domains to create a more complete understanding of emotional processes. By examining both common and unique features across behavioral, physiological, and neural domains, researchers can build a more comprehensive picture of how emotions are represented and processed in the human brain. Despite the criticisms we've discussed, there is considerable evidence supporting the neurobiological underpinnings of basic emotions. Research has consistently found specific neural patterns associated with distinct emotional states. A comprehensive meta-analysis combined findings from multiple neuroimaging studies to examine the neural correlates of basic emotions. This approach provides more robust evidence than individual studies alone. The meta-analysis examined 5 basic emotions: fear, anger, disgust, sadness, and happiness. Each emotion showed consistent activation in specific brain regions across multiple studies. Brain imaging reveals that different emotions activate distinct neural networks. These images show multiple views of the brain with highlighted regions that consistently respond to emotional stimuli. This brain visualization shows the discrete activation patterns found in the meta-analysis. Each emotion produces distinguishable neural signatures that can be reliably detected across different studies and participants. The evidence for basic emotions comes from multiple converging sources: neuroimaging studies, lesion research, behavioral data, and physiological measurements all point to the same conclusion. Discrete emotions have consistent and discriminable neural correlates. This converging evidence provides strong support for the neurobiological underpinnings of basic emotions. The consistency of findings across different research methods strengthens the case that discrete emotions are real and measurable phenomena in the brain. After examining the extensive research on basic emotions, we arrive at a nuanced conclusion about this fundamental question in neuroscience and psychology. The debate about basic emotions is far from settled. While critics have raised valid concerns about traditional approaches, the evidence supporting neurobiological foundations of basic emotions remains compelling. Dismissing basic emotions entirely would be premature. The neuroimaging evidence, combined with lesion studies and research on emotional awareness, provides a robust foundation for understanding discrete emotional systems. However, our understanding must evolve. The complexity of emotional processing requires moving beyond simple brain region to emotion mappings toward more sophisticated network-level analyses. An updated framework must incorporate network-level analysis, consider pluripotentiality of brain regions, and integrate multiple types of evidence from neuroimaging, lesion studies, and behavioral research. The concept of basic emotions can still be fruitful if updated to reflect current neurobiological knowledge. Future research should focus on understanding how emotional networks operate dynamically and how different brain regions contribute to emotional processing in context-dependent ways.